Everything you were taught about the origin of life is built on one assumption. That assumption is sunlight. Life needs the sun. That is not a theory. That is the axiom the entire edifice of biology rests on. Photosynthesis feeds the plant. The plant feeds the animal. The animal feeds everything else. Remove the sun, and every living thing on Earth dies. This is not controversial. This is the first fact you learn. In 1977, a submersible descended into a section of the Pacific Ocean floor where no light had ever reached. What they found there did not just challenge that assumption, it dismantled it. 2 miles below the surface, in complete and permanent darkness, in water hot enough to melt lead, surrounded by chemicals toxic to almost every form of life on Earth, something was alive. A lot of somethings, and none of them had ever needed the sun. To understand why this discovery was so disorienting, you need to understand what was believed before it. For the entirety of recorded scientific history, the food web was understood as a pyramid with one foundation: photosynthesis. Sunlight strikes chlorophyll. Chlorophyll converts it to sugar. Sugar feeds bacteria. Bacteria feed plants. Plants feed everything else. The chain is long and complex, but the base is always, without exception, the sun. The deep ocean, by that logic, should be nearly barren. Light cannot penetrate below about 3,300 feet, roughly 2/3 of a mile. Without light, no photosynthesis. Without photosynthesis, no base to the food chain. What lives there should be sparse, slow, and wholly dependent on the slow rain of organic material descending from above. Dead plankton, whale carcasses, the debris of a sunlit world drifting down into the dark. The deep ocean was not expected to be empty. It was expected to be a desert, a cold, dark, almost lifeless desert. In February 1977, a research team aboard the vessel Nor was investigating a section of the Galapagos Rift, a region of the Pacific Ocean floor where tectonic plates pull apart. They were not looking for life. They were looking for warm water anomalies. Geologists had predicted that seawater seeping into cracks in the ocean floor, heated by the magma below, should emerge somewhere along the rift as a warm current. They wanted to find it, measure it, and map it. The submersible Alvin descended. At roughly 8,200 feet down, a mile and a half below the surface, in water that should have been near freezing, the temperature sensors spiked. They had found the warm water. Then the pilot looked out the viewport. It wasn't supposed to look like this. Clustered around the base of the thermal vents were tube worms longer than a human arm, giant white clams the size of dinner plates, crabs, shrimp, anemones, fish, an entire ecosystem, dense and teeming, gathered around columns of superheated water pouring out of the ocean floor. The chief scientist on that dive later described the moment as the most profound of his scientific career. He said they had found a Garden of Eden at the bottom of the ocean, a garden that ran on something no garden had ever run on before. The question that hit the research team immediately and has driven deep-sea biology ever since was simple. What were these animals eating? There was no sunlight, no photosynthesis, no debris field large enough to sustain an ecosystem this dense. The answer, when it came, rewrote the rule book of life on Earth. The vents work like this.
Cold seawater seeps down through cracks in the ocean floor, descending toward the magma layer below. As it approaches, it heats. It chemically reacts with the surrounding rock. It dissolves minerals, iron, copper, zinc, and most importantly, hydrogen sulfide. Then, pressure forces it back upward, erupting through the ocean floor at temperatures between 60 and 400 degrees Celsius, carrying a rich chemical payload with it. To us, hydrogen sulfide is toxic. It smells of rotten eggs. It kills humans at concentrations above 100 parts per million. To the bacteria living at the base of the vent ecosystem, it is food. These bacteria, chemolithotrophs, to use the scientific term, do something that no living thing was known to do before 1977. Instead of harvesting energy from sunlight, they harvest it from chemical reactions. They oxidize hydrogen sulfide, stripping electrons from it, using the released energy to convert carbon dioxide into organic compounds. The process is called chemosynthesis. It is photosynthesis without the photo. Light replaced by chemistry. The sun replaced by the earth itself. The bacteria form the base of a complete self-sustaining food web. Some free float in the vent water. Others form thick mats across the surrounding rock, visible as white or orange films coating the sea floor. Others live inside the bodies of the animals themselves. The tube worms, Riftia pactyla, are among the most extraordinary animals ever discovered. They have no mouth, no stomach, no digestive system of any kind. They do not eat. Inside their bodies is a specialized organ called a trophosome, a dense mass of tissue that can account for more than half the worm's body weight. The trophosome is packed with chemosynthetic bacteria, billions of them living symbiotically inside the worm. The worm absorbs hydrogen sulfide from the water through a feathery red plume at its tip, delivers it to the bacteria, and the bacteria produce the organic compounds the worm needs to survive. The worm is not an animal that hosts bacteria. It is a biological housing unit that evolution built around bacteria. The distinction matters.
Riftia pactyla grows at a rate of up to 33 inches per year, among the fastest growth rates of any known marine invertebrate. How an animal with no digestive system generates the energy to grow that quickly on bacterial chemistry alone is still not fully explained.
The clams do something similar. Their gills are packed with chemosynthetic bacteria. The clams absorb sulfide through their foot, which they press directly into the vent sediment, extracting what the bacteria need. Their blood contains a unique form of hemoglobin, a variant evolved specifically to carry both oxygen and hydrogen sulfide simultaneously without the two interfering with each other. A problem evolution has not solved anywhere else on Earth. The clams solved it. The vents themselves are not subtle. Black smokers, the most dramatic type, erupt superheated water at up to 750 degrees Fahrenheit. The water would boil instantly at the surface, but at this depth, the pressure is so extreme, 250 atmospheres, that the water cannot form bubbles. It stays liquid at temperatures that would vaporize it anywhere else on Earth. The physics that governs water at the surface simply does not apply here. The dark plumes that give black smokers their name are not smoke. They are mineral particles, iron sulfide, copper sulfide, zinc sulfide, precipitating out of the superheated water as it hits the near-freezing deep ocean. The particle cloud billows upward and outward, and over years and decades, the minerals build up into towering chimneys.
Some of these chimneys stand nearly 200 feet tall, structures taller than a 15-story building, built entirely from the chemistry of the Earth's interior, rising from the ocean floor in permanent darkness. They are, by any reasonable definition, one of the most extraordinary physical structures on the planet, and most people have never heard of them. The tallest known hydrothermal chimney, discovered on the Juan de Fuca Ridge, stood nearly 150 feet tall before it collapsed in 2000. The rate at which these structures grow and the internal chemistry that keeps them structurally stable is still being studied. We do not have a complete mechanical model for why some last decades and others collapse within years. Since 1977, more than 700 vent sites have been discovered across the world's ocean floors. Each one is an island, separated from the others by miles of cold, nutrient-poor abyssal plain. Each vent community is effectively isolated. Species cannot easily travel between them. Evolution acts on each population independently. The result is a biological laboratory unlike any other on Earth. Shrimp that have no eyes but can detect infrared radiation from the vent water. Crabs coated in bacterial fur. Strands of chemosynthetic bacteria so dense they look like a white fleece. Octopus that have colonized vent environments and appear to have adapted their thermal tolerance in ways that other octopus have not. Snailfish found living at vent sites deeper than any previously recorded vertebrate habitat. And all of it running on chemistry, none of it dependent on the sun. By the mid-1980s, the scientific community had absorbed the discovery of chemosynthesis. It had been classified, documented, peer-reviewed, and filed. And then, a small number of researchers began asking a question that the original discovery team had not. Not what lives at these vents, but what if life started here? The origin of life on Earth remains the deepest unsolved problem in all of science. We know when it happened, roughly 3.7 to 4.1 billion years ago, within a few 100 million years of the planet's formation. We know what it produced, the first self-replicating molecule, most likely an early form of RNA, capable of storing information and catalyzing its own reproduction. What we do not know is where or how. The traditional hypothesis, Darwin's warm little pond, imagines life beginning in shallow, sunlit water, where organic molecules concentrated and combined in the heat. Lightning, UV radiation, thermal cycles, simple chemistry gradually becoming complex chemistry, complex chemistry gradually becoming something that could copy itself. It is a reasonable hypothesis. It is also, increasingly, a contested one. In the late 1980s, a geochemist named Michael Russell proposed a different origin story. Not hot acidic surface pools, but cool alkaline hydrothermal vents on the ocean floor. Russell's hypothesis begins with the chemistry of early Earth. The young planet had an ocean, but almost no oxygen. The atmosphere was a mix of nitrogen, carbon dioxide, and methane. The ocean was mildly acidic, and beneath it, the ocean floor was rich in a mineral called olivine, which reacts with seawater through a process called serpentinization, producing alkaline, hydrogen-rich fluid that seeps upward through the rock. Where this alkaline fluid met the acidic ocean water, a gradient formed, a chemical gradient, a proton gradient to be precise. And here is where the hypothesis becomes extraordinary. The proton gradient is not a detail, it is the engine. Every living cell on Earth today, from the bacteria in your gut to the neurons firing in your brain right now, runs on a proton gradient. It is the fundamental mechanism by which cells generate energy. The mitochondria in your cells create a proton gradient across a membrane, and the flow of protons through that membrane drives the production of ATP, the molecule that powers almost every biological process in your body. Russell's argument was this: the first living systems did not need to invent the proton gradient. It was already there, built into the geology of the early ocean floor. Life did not create its power source, it found one that already existed and learned to use it. The implication is vertiginous, the mechanism at the core of every living cell on Earth today, the proton gradient that powers your heart to beat as you watch this, may be a direct inheritance from the chemistry of a hydrothermal vent 4 billion years ago. Your mitochondria may be a memory of the ocean floor. In 1995, the Hubble Space Telescope photographed Europa, one of Jupiter's moons. The images showed a surface of fractured ice, ridges and cracks suggesting movement, a moon whose frozen exterior was shifting, grinding, rearranging. The implication was immediately understood by planetary scientists. Beneath the ice, there was liquid water, an ocean. Europa's ocean is kept liquid not by sunlight. Jupiter's light is 50 times weaker than Earth's, but by tidal flexing. Jupiter's immense gravity continuously squeezes and releases the moon, generating heat through friction in the interior rock. The same heat that powers Earth's hydrothermal vents is generated on Europa by an entirely different mechanism with an identical result. Liquid water, geological heat, chemical gradients. The three conditions that the hydrothermal vent hypothesis identifies as sufficient for life. Europa has all three, and Europa is not alone. Enceladus, a moon of Saturn, erupts geysers of water vapor and ice particles from its south pole. NASA's Cassini spacecraft flew through these plumes and detected hydrogen, carbon dioxide, and methane, the chemical signature of active hydrothermal venting on the ocean floor beneath the ice. Not a theoretical possibility, an active, confirmed, ongoing process on a moon of Saturn, right now. Titan, Ganymede, Ceres, Pluto. Planetary scientists have identified liquid water ocean candidates in each. None of them close enough to the sun for sunlight to matter. All of them potentially warm enough, chemically active enough for the vent hypothesis to apply. Before 1977, the search for extraterrestrial life was, in practice, a search for planets with sunlight and liquid water at the surface. That search had one primary candidate, Mars, in its ancient past. After 1977, the search became something entirely different. It became a search for heat gradients and chemical energy in the dark. And suddenly, the solar system was full of candidates. But we have not yet sent a probe to penetrate Europa's ice and sample its ocean. The mission, Europa Clipper, launched in 2024, will orbit and study the moon, but will not break the surface. The direct question of whether Europa's ocean contains life remains, as of today, unanswered. We may be within a generation of answering it. In the 47 years since the Alvin dive, the discovery of hydrothermal vent ecosystems has changed 5 separate fields of science. It changed biology by proving that sunlight is not a prerequisite for life. It changed geology
by revealing that the ocean floor is not inert rock, but an active chemical system in constant exchange with the water above it. It changed astrobiology by expanding the definition of a habitable environment from a planet in the Goldilocks zone to anywhere with liquid water and a chemical gradient. It changed evolutionary biology by providing a credible environment for the origin of life that requires no sunlight, no lightning, no warm little pond. And it changed our understanding of what is possible, not as a philosophical statement, as a data point.
The tube worms living at hydrothermal vents today are, in a sense, our oldest relatives. Not in the way that all life shares ancestry, though it does, but in the more specific sense that they may live in the same conditions, run on the same chemistry, exploit the same gradients as the thing that started all of this. They may be the closest living analog to life as it was 4 billion years ago, before eyes, before lungs, before neurons, before anything we recognize as an animal, before the sun mattered at all. We have visited the moon 6 times. We have sent 29 missions to Mars. We have launched probes to the edge of the solar system. We have explored approximately 25% of our own ocean floor. In that 25%, we have found 700 vent sites. In the 75% we have not yet seen, the mathematics suggest there are thousands more. Each one a self-contained ecosystem. Each one running on chemistry, not sunlight. Each one potentially home to species we have not yet named. The 1977 discovery did not just add a new chapter to marine biology, it changed the question. For all of human history, the question was, where else in the universe could life exist like it does on Earth? Since 1977, the question has been, where else in the universe could the conditions exist that we now know are sufficient? The answer, as best we can determine with the instruments and knowledge we currently have, is almost everywhere. 47 years ago, a submersible pilot looked out a viewport 2 miles below the ocean surface and saw life where there should have been nothing. The universe has not looked the same since.